Hey, hi everyone. Welcome to today's session. I think before we start, there's just some minor glitches with the video that we're trying to fix. But in the meanwhile, I think we can get started. Welcome to the session today. My name is Roohi. I'm just moderating and introducing our speaker for today. But before I do that, I would like if we can use the chat to just talk about, just introduce yourself, where you're from, and what you're hoping to gain or learn from the session today. So it'll help us understand, your expectations from the session, and it'll help us to get to know you better.
Okay, go ahead, Rui.
Yes, hi Manu. Sorry that you had some trouble joining in, and I'm glad that worked out. So, hi everyone. Welcome to the session again today. We're delighted to welcome Manu Jain Goyal from the International Journal of Cancer as our speaker today. Manu's main role at IJC has been to ensure the integrity of research that's published in the journal by checking relevant issues such as plagiarism, paper mills, the use of AI, image falsification, and cell line authenticity. And all of these issues, as we know, are fundamental to maintaining the credibility and the trustworthiness. Of course, it is important that all parties involved in publishing, all the stakeholders should adhere to the principles of integrity to achieve high quality scientific publications. And I think that's exactly what Manu will be discussing in today's session to talk about good scientific practices and research integrity. I think we all have a lot to gain from her editor's perspective. So yes, Manu, the floor is all yours. You can start, and thank you very much for taking the time out for this.
Thank you, Ruhi, for introducing me, and thank you, Mary, for giving me this opportunity and ease to talk about good scientific practices and research integrity. And thank you all for joining me today. I'm working at International Journal of Cancer as a scientific advisor. International Journal of Cancer is officially a journal of Union International Cancer Control, and it covers a wide range of topic which are related to experimental and clinical research. And Professor Dr. Christoph Plass is a editor in chief of our journal, who is also a professor in DKFZ currently. And we receive submissions over 3,500 every year, and the acceptance rate is 13%. We are a team of 7 people. So Dr. Franca Bianchini is a managing editor, and Mrs. Yvonne Owl, who is a deputy managing editor. Then we have 2 editorial assistants, Lisa Brown and Dara Koroleva. Then we have a research integrity team of 3 people right now. There used to be 4. So right now we are Dr. Stephanie Heck, Dr. Constantina Falida, and myself, and Dr. Nicole Soren, who was in our research integrity team and who started basically the cell line authentication at IJC. I will be talking about this in my later slides. And Dr. Stephanie Heck, and Dr. Nicole Soren both started basically research integrity at IJC. So they are majorly involved in this part. And we joined IJC, me and Constantina, and followed their path basically. So yeah. So as I said, and also as Ruhi mentioned, that IJC is a pioneer in the field of data integrity. We follow a strict policy of the data integrity and pays particular attention to preventing potential scientific misconduct. So I guess everybody knows the basic definition of the good scientific practice, which basically involves three basic principles: transparency, which means that there should be a reproducibility, and if the data has been if the data has been used, the source should be disclosed. And to maintain its integrity, we control the quality of the integrity, and we control the quality of the data, and then and the author should take the responsibility of their data. And then the third one is the FAIR principles, where the data should be accessible and reusable. So we strongly recommend authors to submit their data in the data repositories, and it should be not just deposited, it should be accessible and reusable if somebody wants to have an access. So we have certain quality control steps to ensure reliability and reproducibility in IGC publications. So the first step we do is a plagiarism check using Authenticate. So basically to see if there is if it's a suspicious paper or there is overlap present in the manuscript. Then we do image checking, and we do ask for raw data. Then, as I mentioned earlier, the cell line authentication, it's very important for our journal, especially because we do receive papers based on molecular studies and cell biology studies. And for those kind of studies, cell line authentication is very important. And it is very important to know that, that you are using the correct cell lines. And yes, the data availability statement, meaning the author should take the responsibility of the data presented in the study. And if it has to be submitted in the repository, then it should be submitted. And then we do also ask for sequencing coverage and quality statistics, meaning if there is a omics data or next generation sequencing data, authors should be able to provide the quality statistics of their data in order to judge whether the quality of their data is enough for the publications. And author contribution statement this is also a very important one, because we need to know the authors that are mentioned in the list, that how much they have contributed and in what manner they have contributed. So it should be stated in the author contribution statement. Then the last but not least, the large language models and AI tools. So we have recently updated our guidelines, that we do not accept the use of AI tools. I will be talking more in detail each and every step of quality controls. So let's start first with the plagiarism check. We use Authenticate to check plagiarism. Here you can see one of the example that we see that the total overlap is 66%. When we see that the overlap is very high, this could mean it is a suspicious paper. And not always, but in many cases, this means that it could be a suspicious paper. When we see that there's a high overlap, it's very important to know that what kind of overlap is present and where the overlap is present. And when the statements or the sections are taken, then they should be cited, and they should be cited, and if they are, no matter if they are from the same authors or from the different authors. But it's a good scientific practice that you need to include the citation. And also during the Authenticate run, sometimes we do see that there are overlaps present in the figures that they pop up in this Authenticate report. For example, and then we see, so when we see all these things are present there, then we check if it's if it's a paper mill paper. And then we do also check for paper mills. And so these are the very common features of classical paper mills paper, which contains like similar structured titles, and then the authors do not have institutional emails, and then maybe logical or very unsophisticated study approaches. And sometimes you will see the texts are recycled and the layouts are very similar. And then the data is fabricated, or it's used from stock images. And yes, as I mentioned again, that as I mentioned earlier, and I'm mentioning again, that it is that they authors are reluctant, that they do not want to share the cell line authentication documents, and also do not want to share their data. So like, for example, here you see what I'm talking about for the classic paper mill collection. So this is the paper which was submitted to IJC. And here you see the figure layout, which are similar to two other manuscripts which were already published in Oncology Report and International Journal of Molecular Medicine. And then when they popped up an Authenticate report, and do you see that they have similar layout, similar presentation. So this is a very typical example of paper mill collection. Then in the next row, since we are receiving a lot of biomedical research papers, we do receive a lot of manuscript which do contain images. So to find out if the images are duplicated or have overlaps, we are using image twin software, which is AI based software to detect issues in figures of scientific articles. So here, for example, you can see in these two figures, this figure is from the main figure, and this is from the supplementary figures from two different cell lines, but they do contain very similar, highly similar microscopic images. So this is not acceptable. This is clearly a misconduct. And then in the next slide, for example, here you can see that the tissue sections and different tissue sections, and then in different conditions, and then you see that that the overlaps are present in different conditions in figure 3 and figure 5, which is also not acceptable. You can't use the same images or overlapped images for different conditions until unless it is declared, or for same experiments, if it has been used, it has to be declared. But this one is not acceptable. Then this is the image for a Western blot, where protein expression is detected using this technique. And then this was detected using image twin software, and then further to confirm that this is a real duplication, then we analyze this image using Adobe Photoshop. So here you can see on the top panel this Western blot band and this Western blot bands. They are flipped and converted, but they are duplicated Western blot bands. So this is also not acceptable. This is clearly a misconduct. For a different conditions, for different proteins, you can't. It cannot be true that you have same Western blot band patterns. And as I mentioned earlier, that we do ask for raw data, whether it is a Western blot or a microscopic image, or so meaning, for example, here, if this is a presenting figure, main figure in the manuscript, then you need to provide a clearly labeled, uncropped Western blots as a raw data. So for example, here you can see the full membrane of the Western blot, and these proteins are labeled here, which is fine. But what is not fine is that they are not labeled like the samples which are labeled here, and also the molecular weight. So it has to be clearly labeled for which figure, for what sample, molecular markers, for what protein, uh, and should be provided as a raw data. Then if you have microscopic or electron microscopic images, or again, if you are using only a part of the whole image, a small section, you need to provide a complete image from where the section was taken, and you should highlight it. And again, you see it's not labeled for what it belongs to. So this is not acceptable. And the original image should include the scale bars, which you see here, left on the bottom. And yeah, sometimes we do receive manuscript where we ask for raw data. They would just provide the raw data, the similar image in a enlarged as an enlarged image. No, this is not fine. You need to provide the uncropped image as a raw data. Yes, so as I mentioned earlier, that we do receive a lot of biomedical research paper which contains cell line studies. So for this, it is very important to know that the correct cell lines are used. For example, here you see there are two cell lines, HIP2 or cell lines and INT407 cell line. So HIP2 cell lines originally believed to originate from laryngeal carcinoma, and INT407 believed to be embryonic intestine cell lines. But later on, it was found out that both were contaminated with HeLa cell lines. So basically, they are HeLa cell lines, not laryngeal carcinoma or embryonic intestinal cell lines. So since 2010, IJC is the first journal who started requesting proper authentication of all human cell lines which are used in a manuscript, and they should not be, the authentication document should not be older than 3 years. So even if the cell lines are new cell lines are generated in the labs, or there are subclones which are generated, we need proper cell line authentication report, which should not be more than 3 years old. So Dr. Nicole Soren and IJC team published an article in AMBO Journal on the cell line authentication, and so because we do strongly believe that every journal and every publisher should follow and ask authors to provide the cell line authentication report. And last year in EASE 17th conference at Istanbul, Dr. Konstantina Falida, she presented a poster on cell line authentication quality control, and she won the first prize for that. So this poster and video is available on EASE website, if I'm correct, Mary. This video is also available. So if you want, you can go and watch the video on there. So yeah, as I said, that IJC requests authors to share their data, which supports the study, and it should be it should be replicable when you are presenting the data. Therefore, author should share the data. And when omics data is used or analyzed or generated, or next generation sequencing data, if there is a computational study, then for that you need to provide source code, or if it's a machine learning study, then for that also we need codes, and also imaging data, if it has been used, they should be available in a public repository. If it is a sophisticated data, there are public repositories available where you can make the access restrict. But data and the details should be still available. And if there is no structured public repository available for the data, then it can be provided as a supplementary material or it can be made available upon request. Yes, as I already mentioned that we do ask authors to provide sequencing quality and coverage statistics if omics data or next generation sequencing data is present in the manuscript, because it is very important to judge whether the quality and coverage of the next generation sequencing data are sufficient to support the conclusions. And in the author contribution statement, it should be explained the contribution of each author involved in the manuscript, preferably according to the credit taxonomy. And for the AI tools and large language models, we do not accept, IJC do not accept if any kind of images and graphical elements are produced using these tools. This is not permitted. But in any manner, these tools have been used like for grammar or for grammar correction or spelling correction. It should be acknowledged and should be described in detail that which kind of tool, which version was used. And these tools do not qualify for the authorship because they are not capable of taking responsibility. So humans should be responsible for for presenting their data and whatever it contains in the manuscript, but tools cannot be qualified as an author. Yes, so we do. So these were about the quality control steps and about the IJC guidelines, what we have. But yes, we do receive alerts from anonymous whistleblowers and readers once from the old published manuscripts. So there is an online journal club, it's called Pub here, where users can anonymously comment on scientific papers and do notify and do comment about the falsified data if it is present in the manuscripts. So when we get the alert, we do follow up on alerts of misconduct in published articles. And when we do post investigation, and then IJC follows copy guidelines for handling the research misconduct. And there is recently copy updated handling of paper mills also, which is available online on the copy website. So there are three kinds of corrections which are right now, I think everybody follow it. First is this erratum, where when there's a error present, and fixing this error should not affect the conclusion of the study. Then we do publish erratum. But in case there's a serious misconduct is present and it can change the conclusion of the study, or it's invalidate the conclusion of the study, then paper has to be retracted. And then there is another type of correction which is called expression of concern, when it is used to used in unresolved cases where the evidence of misconduct is inconclusive, meaning that when you are doing investigating and if you think that you need raw data, but the raw data is not available, and you still have the doubt about the data which is presented in the manuscript, in that case we do publish expression of concern. Yeah, and I find these links as a useful resource, as I mentioned about the COPE already, which is a very useful resource. They have Copy Forum and several other several guidelines to handle the research misconduct and paper mills. And recently, EASE also launched a EASE Forum where you can discuss about your case, and also if you have any doubt you can ask questions, and people are really open to discuss. And you can also do it anonymously if you want. But yes, definitely these are very helpful in case you are looking for a guidance. And then yes, there's a retraction watch. I think everybody knows about it. They do publish articles about retracted papers, which I find articles very interesting. And yeah, and there is this uh, database. It's called Salosaurus database, which is for the saline database, where you can, if you want to know the saline you are using is the correct saline. So first of all, if you just want to know in general about the saline, you can just put the name on this database and you can find out if there is any problems or any contamination was found out. But definitely what you are using the cell line, you need to do the authentication for these cell lines, and then you can compare your report which are present on this database. Then you would know whether your cell line is correct or not. Yes, and the recently Retraction Watch published an article on their where Frontiers retract nearly 40 papers which were linked to authorship for sale. I found this article very interesting if somebody wants to read. And yeah, so this I attended maybe last month or a couple of months ago from Ivan Oransky, where he also mentioned that awareness has increased, where like people know that they do comment about the research misconducts, and journals are also taking it seriously, and retractions are happening, but they are not enough, which is true. We need improved process for retraction. So yeah, so this is another interesting article from Retraction Watch. Yes, so the take-home message would be for authors. Please do follow good scientific practice to ensure reliability and reproducibility. And for readers and authors, if you see irregularities, if you spot any sign of misconduct in the published paper, please do raise your concerns. You can anonymously comment on Papier, or you can directly email to journal, but so that they can follow up and do the investigation. But do say something. And as an author also, I would say, if you know that that you have found an error in your own paper, it could be an honest mistake, honest error. But please do reach out to correct that mistake. And I read, and I do also believe that that correcting a mistake should not be uh, should not be should not be taken as a back step for your career. It should include it as a healthy practice in science. So and it is I do believe it is very important to uh, to to uh, to uh, uh, to notify and to speak about the misconduct. Yes, and for journals, journal guidelines should be updated. Be consistent with the quality control steps. Should not make exceptions for anyone, no matter from where the authors are. The quality control steps should be similar to each and everyone. And implement, if your journal receives studies which contain cell lines, please do ask for cell line authentication report. It is very important, because if somebody is working on HeLa cell lines, but if somebody is working on gastric cell lines and it is contaminated with HeLa cell lines, it is not a gastric cancer cell line study. It is not a gastric cancer study. It is a cervical cancer study. So please do ask for authentication reports. And when you asking for data sharing, please do make sure that data is truly available, because in my experience, we do receive manuscripts where we ask authors to submit their data. They would provide the link, but when you open the link, oops, sorry, there is nothing. The data is not exist. The data does not exist, and sometimes the details are available, but the raw data files are not available. So you need to know what authors are providing, what kind of things, what kind of data they are uploading. We do not need processed data, we need raw data. So if somebody needs to reanalyze, reproduce, or do further studies, they need to have access for the raw data. And if any kind of misconduct is present, or or you got an alert, or somebody is reaching out to the journal or publisher, take action against any misconduct. So yeah, this was from my side, and I'm open for the discussion.
Thank you, Manu. I think this was very, very useful. I mean, I'm sure we all learned a lot from it. I know I did. I would request everybody here to please, you can unmute if you have a question. Keep your cameras on, and we can have more like a discussion or an informal conversation with Manu, instead of the more formal way of typing in your questions. Just keep your camera on and ask your question, and we can take it from there.
I think there is one question from Anita.
Yes. Does IJC run checks for text content generated by AI or generative AI? And if yes, which tool does IJC use?
So yeah, we did use we until recently we were using GPT detector, but I think people are getting very smart with the AI. They do. It's not easy to detect AI generated text, but recently, STM Integrity Hub, they have developed a tool, paper mill detection tool, which I know it also contains one of the feature where AI generated or driven text can be detected. But right now I do not have access to that tool. But yes, definitely we do need a robust tool to find out if the papers are AI generated or text is generated by AI or fake papers, basically.
Yes, because Manu, just to follow up, I read some posts on how some of these tools that claim to detect AI-generated content may not be very reliable also, and that sometimes puts the authors in a spot, right? When the journal comes
Right.
back saying what has happened, and
Yeah.
I guess not all tools have been validated for their accuracy and reliability. That's where my question came from. As if I may ask one more question, I know that some journals have a default statement in the template where authors can declare the use of a generative AI tool for maybe outlining the text, and then they say that I built on the draft and I wrote everything else myself. Does IJC have something like that in place in the template?
Yes. We do have a section in questions that if any kind of editing, outside editing was used, or any help was used. But we are also going to have a new update in our system where there will be a specified AI question will be there if somebody has used any kind of AI tool and where and which one. So yes, definitely it's going to happen soon. But in our guidelines, it's already there. But when in the submission process, it's going to be integrated soon.
Thank you.
I had a related question. You spoke about AI tools like image twin softwares for detecting issues. So, do you have a view on how accurate these tools are for
Yeah,
image
see,
detection?
these tools are very helpful, but we need to human needs to check it, right? Because these tools can also have false positives, right? So they can give you several results, several overlaps they will give you, but you need to recheck by yourself. Like as I mentioned in one of my slide for the Western blot, that after running this image tint software, we do check also using Adobe Photoshop, and there we have extra plugins, and using that we do confirm that if it is the real application or not. So, but in most of the cases, I would say it's very reliable and helpful. Yeah.
What about the role of like, say, when you talk about review, you also mentioned raw data, and you said how you ask for the raw data to be made available. So are peer reviewers with your journal also expected to go through to verify that raw data and to confirm if that data is correct, or is that outside the purview of the peer reviewing responsibility?
No. So when we have reviewers on board, they have these several questions while they are submitting. So they do. So they need to provide the statement for that if the data which is presented in the study, if it's adhere to this manuscript or not. So that's why authors need to provide an access before sending out to peer review to the raw data. So they do check and then they do comment about it.
Are
And
there any
also,
other questions? Sorry.
sorry.
Sorry, please complete.
No, that's what I was saying. Yeah. Also, sometimes our editors, associate editors, they are scientists. They are all still running their labs, so they also know about the data, and they are also very much updated in their field. So when they are reviewing the manuscript, so they also check if the data what the authors have provided and the details, if it's correct or related to the study or not, and then also reviewers. Yes.
Are there
Hi.
any other questions?
Yes. Can you hear me or see
Yeah.
me? Hi. I'm a freelance copy editor, and I have a question around best practices for a freelance editor when it comes to research integrity matters. Um, as a freelancer, we don't have access to a lot of tools. So, are there any free tools available for detecting plagiarism or image duplication, or what can a freelance editor do in this scenario?
Unfortunately, I do not know if they are if these two, if for example the image twin is not freely available. You need to subscribe for it. And also for Authenticate, I'm not sure, but I don't know if there are any other tools which are freely available. But what I can say and suggest that as a freelance editor, you can always first check, as I said, that if the authors are providing the institutional emails, and then if they they have any peer entry and if they have any kind of papier entry, so like what kind of allegations are, so we need to be sure that the allegations which are there, are they real or valid or not. And then you can always do background check when they are presenting the data, if they have published in their in the past on the current topic or not. And yeah, also I think ORCID ID, I would say ORCID ID can play an important role. I should have mentioned earlier that IJC also asks for ORCID ID, and then within the ORCID ID, usually the details of the authors are available, and you can see in the past in the professional years what kind of studies they have done and what kind of manuscripts they have published. So these are the basic checks which you can do. Yeah, but unfortunately, I'm sorry, I cannot say anything which are available freely available for the plagiarism tools. Yeah.
Thank you so much, Manu.
Maybe we can open this question up to the audience, and
Yeah.
if there's anybody else here who has a view on this and has used a low cost or a free tool, please share it with us now.
Yes, so Nana, I have a related thought on this. The issue that you have raised, you also need to consider whether these things come under the scope of work that a copy editor is doing, because the article or the paper that has come to you for editing, it would have come after peer review.
Right.
So all these checks would have happened. But the scenario which you are discussing, it will arise when you are dealing directly with the author.
Exactly. Yes. That is why I'm asking.
And I think that's a situation or scenario that Cactus deals with. So Roohi, you have any inputs on that, like other than what Manu has already told us?
Right. No, I think at Cactus there are some teams. I think they use Authenticate, but or the plagiarism checkers that are available. But aside from those, I'm not aware of any myself.
Okay. So Nana, like in the book that we have just published a few months back,
Yes.
Discover the Editor in You. So
Right.
there is a resources section towards the end of the book where you will have a listing of some free tools or software that you can use for checking plagiarism and for a lot of other things that a copy editor needs to do, whether it's like checking your references or doing fact checking. So those pages will be very useful to you. If you have the book, you can check them after this session, or if you don't have the book, you can order the book.
I will. Yes.
Yeah.
I will. Thank you. Vivian.
Okay.
I just wanted to add to that, Sunaina. I agree with that. I think as freelance copy editors, we do not have access to institutional resources.
Yes.
And sometimes, or many times, it's not within the scope of what we are expected to do. If it is, and then we're working with an institution, like sometimes I've worked with an academic institution, and then their libraries usually have an institutional copy of Turnitin or IThenticate.
Okay.
And if you're working with a collaborator within a university, that's a resource that you can use. If you don't have access, sometimes your collaborator can run the test and the check within their institution.
Yeah.
And they can share the report with you. So that's one other thing that's one option that I've used sometimes if required. The slight challenge with using the free resources is sometimes you have to upload the text, and when you're working with confidential text with your clients,
Yes.
it is it's not an easy decision to make on whether you want to upload it. So that's something I just want to say.
Thank you.
Manu, I had one other question, if it's okay. It's probably a subjective one. How many of these, suppose you find, given the issues you find that you flag or raise with authors when you run checks for integrity and ethics, what percentage do you think are deliberate, and what proportion do you think may be lack of information or ignorance? I'm just wondering here, is there some kind of educational effort that can be made at some other level, right? Not at the probably at the stage when they're submitting. So just wondering, what do you think about how much is deliberate versus how much is a lack of knowledge or just ignorance?
So first of all, when we see the sign of any suspicious, and the editors see if that it's a suspicious papers, and also if it lacks the novelty of the study, so we do not proceed further. It is rejected right away. But if it still goes, if it is decided to send out for peer review process, then we do perform checking before sending out to review. And if we found out any evidence, of course, you need to have evidence, then we reject. So I would say when we have suspicious papers, most of them, I would say they are rejected right away. They are not uh, selected for review process. I don't know if that was your question or. Hello?
Yes, yes. I see. No, I was just wondering whether you think that, but that's probably something you may not have information on whether they intentionally submitted such information or wrote it that way, or whether it was something they didn't know about, maybe.
Yeah, I mean, I do not have such a statistics right now, but only one thing cannot tell that if this was intentional or a fake paper. If you do see all together all the signs that they could be product of paper mill or fake paper, then you could say. But I do not have a statistics on that right now. But yeah, I'm sorry, I don't know if I can give a specific answer on that.
No, sir. Thank you. Or if you have any suggestions on a first or second.
Sorry, I cannot hear you.
Does copy have that?
Hello. Sorry, I couldn't hear your question completely.
No, I was just saying, is there a resource that maybe copy has some resources that early career researchers can use to find out how to adhere to best practices?
Yeah. So for the as I mentioned earlier, in one of my slide, that the typical features of the paper mill venue. So for us is that, like, first what we check is we do the authenticate run, right? If we do see authenticate run is higher, and then these citations are popped up in the overlaps, then you have to see what kind of overlaps and they are presented. Then you will also see there are several papers which are published in they are presented in a similar way, and you will see that the title has been changed a little bit, but the figure layouts are same, and you might find out some graph plots are presented in the same way. So these are typical features which you see. But yeah, definitely, if you go on COPE website, COPE forum, or Retraction Watch, and also EASE, I think also has literature on that maybe. So if you can go there and read about it, so I think you will find the best to follow the best practice. I think the COPE would be the best, I would say, to read about it, to follow the best practice. And that's what we also follow.
Thank you very much.
Thank you, Manu. I think this was very useful, and I'm sure there are others here also who have many questions. If you do, you can please send those in, and we will try and see if Manu has the time to take some of those questions offline. But otherwise, I think we've nearly reached the end of this session today, and I would just like to thank everybody for attending today. And Manu, thank you very, very much for taking the time out to do this. We do have such sessions about every month, and if you are so you should just keep in touch with what is happening, stay updated, and sign in for any more of these monthly sessions on a regular basis. I think it helps to stay connected and to learn continually. And I learned so much in today's session also. So thank you very, very much. And of course, if you have any feedback, anything that you feel will help you, please write in to us. I think Mary has already posted in the email address. So yeah, we look forward to hearing from you. Thank you.
Thank you. Thank you, everyone. Thank you, Roohi. Thank you,
Thanks,
Mary.
everyone.
Thank you.
Thank you, everyone. See you next time.